everybody. I'm going to stop sharing for just a moment so you can see our faces. Welcome to the Summer Institute, Module 11. This is Genetic Epidemiology. And we'll be meeting together over the next 3 days to talk about some of the principles in genetic epidemiology. So I'm going to share my screen here. The first session, we're just going to go through sort of an overview for the week and where you can find various resources for the course and give you an introduction to who we are and what we'll be covering. So before I get started, I want to acknowledge that the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Duwamish, Puyallup, Suquamish, Tulalip, and Muckleshoot nations, is the land that the University of Washington is now on. And we're all sitting remotely and attending this workshop from various places around the world. But being that we're housed at the University of Washington, want to acknowledge that this land belongs to other people. So this session, to start off our next few days together, we'll go through an introduction of who we are, talk a little bit about what we'll be talking about over the next few days, and then give you an idea of where you can find various resources. So what types of breakout questions we'll have, how to navigate the Slack channel, the types of activities we'll have during our lectures, and then have a little bit of time to practice with some of this technology. I know most of us have gotten very familiar with it over the last 2.5 years, but just to make sure we're all on the same page getting started here. So I'm Allie Fauner. I'm an assistant professor in the Department of Epidemiology. I'm also one of the associate directors of the Institute for Public Health Genetics. And the Institute for Public Health Genetics is an interdisciplinary program. It's housed in the School of Public Health at the University of Washington, but brings together genetic epidemiology as well as the legal, ethical, and social sides of how genetic epidemiology research is conducted, and then how those results are used in society and public health. So you'll see a lot of the topics that I talk about, bringing my own angle, have to do with this, a lens of this ethical, legal, and social implications. So I love talking about this stuff. If you are working in the area, please reach out. I'd love to talk with you about how your research and interest can be viewed and incorporate some of these elements. So the genetic epidemiology that I do is centered around precision medicine. And I integrate not only genetic data, but also proteomic data, and then phenotype information from electronic health records to try and get a better idea of how we can improve targeted medications and interventions to people. So getting the right treatment to the right person at the right time. The current outcomes that I work on are cognitive decline, including dementia and Alzheimer's disease, other outcomes related to aging, and then anything related to medication response. I'll turn it over to Sarah here to introduce herself.
So hi, my name is Sarah Lindström. I go by she, her. I'm an associate professor in the Epi department here at UW, and I have a joint appointment at the Public Health Sciences Division at the Fred Hutchinson Cancer Center here in Seattle. So I am an engineer and a physicist by training. I got my PhD back in Sweden, where I'm from. But I really found my passion in genetic epidemiology, and that's where I've been hanging out for the last 15 years or so. So my primary interest is genetic association studies. So I have been studying a lot of different traits, a lot of different diseases. Most of my work is in cancer. And we have two primary focuses there. One is to understand shared genetic risk factors across multiple cancers. So we do a lot of sort of multi-cancer analysis. And my favorite cancer, though, is breast cancer. So if I'm not doing sort of looking at all cancers at once, I spend a lot of my time studying breast cancer. So I'm going to cover a lot of the things sort of it comes to association studies, gene-environment interaction, GWAS, and also those kind of things. And if you are doing any genetic epi studies now, association studies, and you have questions, I'm more than happy to talk. And we're going to come back and talk about opportunities for you all to talk to me and Ali on an individual basis if that's something that would be helpful for you. And now I'm going to turn it over to our TA, Diane.
Hello, everybody. Can everyone hear me? I know I'm having some internet issues, but just nod. Okay, cool.
Yes, you're good.
So I'm very early in my career. I'm a PhD student at the Institute for Public Health Genetics, and I'm interested in both genetic and environmental risk factors for disease, and I'm really focused on Alzheimer's disease and dementia right now. Specifically, I'm really interested in studying multi-ethnic populations. They have really interesting problems and challenges in terms of statistical genetics, and there are different tools to handle that. So I've used tools like GWAS and admixture mapping to try to elucidate some of those genetic risk factors. And right now I'm trying to integrate some social and environmental epidemiology together with that to better understand the full picture of what's going on. So I'm really happy to talk about any of those things, Alzheimer's disease genetics, or just being a grad student. I definitely understand that. And I know there are a lot of you here today. So feel free to message me on Slack or send me an email if you want to chat. Happy to do that.
And we're really lucky to have Diane with us this week. She'll be helping us a lot with the course, and will also be giving one of the lectures tomorrow based on some of her expertise in polygenic risk scores. So we're going to have 2.5 days together. Our half day is today. And what we'll be covering today is some of the foundational knowledge that you'll need to be successful in the later sessions of the course. So we are assuming that you come with sort of a basic math background. You know how to add numbers together and know basic biology. And that's about all you need to know to be successful in this course. So we'll start after this introductory section. Sarah will give you an overview of genetic epidemiology, including some of the foundational information that foundational strategies for epidemiology generally. And then we'll end the day with genetic variation. So talking about what types of genetic variation there are and how we measure, how we connect what we can see with what is happening in the DNA. Tomorrow we'll get a little bit more into specialized methods. So we'll start the day talking about some of the nuances of genetics that make it both really exciting and really challenging to study. So Hardy-Weinberg equilibrium, linkage disequilibrium, and elements related to human ancestry and principal component analyses. We'll talk about how to do studies next. So types of study designs, talking more in depth about specific association studies and how to interpret those results. And then moving into genome-wide association studies, which is one of the key tools in genetic epidemiology. We're going to end Thursday and Friday with office hours, where you can stop by if you have specific questions you want to talk about, or just come by and listen to what your peers are asking. It's a really nice time to ask questions that clarify some of the topics that we've been discussing, but also an opportunity for you to get advice on some of your own projects and make connections across various areas. And there's a note here that you can schedule time to discuss your own projects, and we'll talk about that more a little bit as well. So Friday, we're going to talk a little bit more about the public health applications and more on the farther down the translational pipeline, starting with some concepts of bioethics and implementation. And then we're going to jump back into specialized methods of genetic epidemiology. So how do we handle rare genetic variants? What about gene and environment interactions? Polygenic risk scores, Mendelian randomization studies. These are all important and powerful genetic epidemiology tools that are good to know about. And then we'll end the day and also our few days together with another office hours. So we'll go through a little bit of resources next. So the first thing to know about is this SISG website. I'm assuming you all know how to access it. Here's the website here, which links directly to our module. And it will include all of the resources that you need, including slide decks from the lectures, which we will upload before each session, so you can follow along. We'll upload links to recorded lectures, So I know some of you are in different time zones or can't make certain sessions. You can always view that material later. And then the recommended readings that we have will be uploaded there in exercises. So this is what that landing page looks like for our module. And if you scroll down from where we see, this is where you would end up if you went to that website. If you scroll down, you'll see a description of our module, and then this course assets on the right will continue to be updated. So today you'll see all of the recommended readings for the entire week. You'll see an overview schedule. You'll see our Slack channel, which we'll talk about in a little bit. And then we'll continue to add these slides. So you can see here, slide for session 3, the slides are uploaded. And before every session, you'll be able to find the slides for that session here. We'll also have breakout activities and engagement activities uploaded there too. So in terms of lecture recordings, we'll be recording all of the sessions. Only people with paid registration will have access to the recording links. And if you have specific requests, if you want closed captions or transcriptions or anything else that would make it better for you to be able to view these recordings, please let us know. We're happy to accommodate anything that will make this process of learning easier for you. The recordings will remain available, I think it's for about 3 months after the end of the session. So they won't be available forever, but they will be available for you to continue to visit, or if you had to miss a session, you can view them later. Like I mentioned briefly, for the end of Thursday and Friday, we'll be holding office hours, and this is a 45-minute session to end every day. This is an opportunity for you to ask us any questions that you might have related to the topic area. You don't have to attend these, but if you have a question, or if you just want to hear what other people are thinking about, or other questions that other folks have, please feel free to come. During this time, we will also be offering an opportunity for one-on-one appointments. So if you hear Sarah or myself talk about a topic and you think, hey, I would really like to talk about my research problem with either me or Sarah, we will have 10-minute slots that you can sign up for to talk about your specific project with either Sarah or myself. And if you want to sign up for one of those times, contact Diane, either by Slack. Actually, Slack is probably the best way to do it, so we keep track of it as opposed to chat. So direct message Diane and she'll help you sign up for a time. She can also help you figure out who the best person to talk to is. So if you have a question, you're not sure if it's better to discuss with me or with Sarah, she can help you figure out who the best person is for that. So as you saw on the website, we have a list of recommended readings. And these are papers that go into more depth in some of the topics that we'll be studying and talking about. So these are optional, but if some of them are more in depth on the methods, others of them give you an applied or opinion view or interpret the method in terms of socially important outcomes. So you'll find a paper for every session, and those papers themselves. So we'll provide the PMID so you can look them up. But the papers themselves in their PDF forms are also available on the website. So we will be using breakout sessions throughout our time together. And these will happen over Zoom. They'll be in real time. There'll be groups of about 4 to 5. And you'll be with the same group of people for each day. So it'll change every day, but you'll have the same 4 to 5 people in each of these Zoom breakout sessions. And it's an opportunity for you to practice some of the topics that we'll be talking about together, and also get to know each other. You all come from very different backgrounds, have different expertise, are working on very different projects. And it's really exciting to think about how these methods can relate to your own studies, but also how other people are working with them as well. So we will assign you to a breakout room at the beginning of each day, and then we'll change the organization of these every day. And if you have any questions, send us a note through Slack and tell us your group number, and we will come in and help you with the specific questions. In addition to, if you have specific requests for us to come and help you with those breakout activities, Diane, Sarah, and I will all be rotating through the breakout rooms. So we'll have a chance to talk with you about the material and about some of the activities that we're doing a little more one-on-one, and be able to answer some of your specific questions there too. And we also ask that you share the results and the discussion, the main takeaways from your discussion with your group on Slack, just so we can see sort of if there's big concepts that we should clarify, and what you as your group conclusions came to. And these activities will be uploaded at the beginning of every day. And then at the end of every day, we will upload the answers for those sessions. So we'll talk about them a little bit in each session live, but if you want to go back and reference how really you were supposed to be doing things, if you approach the question sort of in the classically right way, you can go back and see those answers. So for some of the shorter questions, we'll also do Zoom polling. So we are going to practice one of those now. And let's see, I'm going to pull up this.
You want me to launch it?
Yeah, that would be great. Perfect, thank you. So we're curious about where all of you are coming from, tuning in from today. So it looks like we've gotten everybody. So you can see that, as expected, based on time zones, most of our people are joining from North America, but we do have some intrepid learners who are joining from other time zones. I guess the South America may also be in our time zone as well, depending on where. So I think it's really exciting to see people from all across the world joining in. I think that's one of the really exciting things about having a remote session like this is we can share. Oh, sorry. Oh no. Sorry, someone else shared. Okay. Is we can learn from people across the world and share our knowledge together. Oh, okay. Sorry. That's up. All right. Thanks, Diane. Okay, you all should have gotten an email invitation to join the Module 11 Genetic Epidemiology 2022 Slack channel. The Slack channel offers an opportunity to, let's see, and were you able to see those results? I think so. Okay. The Slack channel will allow for one-on-one direct messaging between participants and instructors, as well as opportunities to communicate with each other. And we will try to upload everything as long as space allows, we will be uploading everything to the Slack channel as well. And we encourage you to ask questions in the Zoom chat, but especially in the Slack channel, because those questions will remain available for everyone to view the answers to at the end of the session. So it's especially for people who are viewing asynchronously to be able to come back and see what the answers were to questions that other folks had in the sessions. And you can go back and see answers to what other people were asking as well. So you're welcome to send us direct messages to ask your question, but if it's a general question that you might otherwise ask in an open session, please write the question in the Module 11 Genetic Epidemiology 2022 Slack channel, and we'll be monitoring that throughout the sessions, as well as throughout the week outside of the sessions, to answer your questions and to help you get the most out of our few days together. If you're unfamiliar with using Slack, you can find information, including how to download Slack, at slack.com. And then for any quick questions, you're welcome to use the Slack chat as well. The problem with the Slack chat is it will disappear from view at the end of the live session. So Slack is better for these sort of long-term content questions, but we will be monitoring both throughout. So before we jump into our first breakout activity, does anyone have any big picture questions on the course or things they'd like to discuss before we jump into our content and our time together? All right, so we will get started with our breakout activity. Our first session breakout activities are listed here. You can also download all of the breakout activity questions for our first day together from the website. So that's a nice way so you don't have to write these down or remember them once this slide disappears. And the instructors will all be sort of traveling through the room, so we'd love to get to know you as well in this process. So Diane, go ahead and send everybody out. All right. Well, thank you all for joining us. I think this will be a really exciting time together. Some of you are brand new to the genetic side. Some of you are brand new to the statistical side. Some of you have been working in this space for a long time and never got formal training in it. So I think it'll be really fun to see how we can fill the gaps in your knowledge and help you move forward in your careers. We look forward to spending the next few days together, and we'll take a short break now and get started again with our core knowledge areas in about 15 minutes.
See you all soon. Okay, great. So let's get started with our second session, which is going to talk about epidemiology, and particularly epidemiology, the concept on things that are relevant to genetic epi. And then I also want to talk a little bit about genetic epi and sort of why we care. Hopefully, since you're taking this course, you do already care, but hopefully I can give a couple of additional insights as well. Before I get started, just a few things. These slides are on the website, so you can download them there if you want to either follow along or if you want to look at them later. And we have also uploaded any breakout room exercises. So the way we're going to do these lectures is that we know that 45 minutes are a lot of minutes and it's long, and especially when we've been doing this for 2.5 days, and you might even have done some other institute modules before. So we're going to try to sort of break this down with, like, we will talk for about 20 minutes or so. We will have a breakout activity. It's either going to be discussion or for you to do some hands-on exercises. And then we're going to come back. So all those breakout activities or exercises are on they're on the website. And so we post them at the beginning of each day for the things we're going to do that day. And at the end of the day, we're going to also upload all the answers to an exercises, so you can go back and look at them if you want. Okay, so let's get started. So I think, epidemiology has become, I think, kind of a trending word over the last couple of years due to the ongoing pandemic. And I think, we might not thought ourselves as being cool a couple of years ago, but I think that we are pretty cool as epidemiologists right now. And really sort of what we're trying to do is to try to understand how diseases and other traits are sort of being spread, or how they are sort of showing up in our populations. So I think that epidemiology is obviously a lot of complicated. It's very complicated, and sort of we can see some things in special populations, perhaps not in others, and sort of try to make a sense of all these things are one of our main goals with what we're doing. So, so sort of to speak on that, really the major goal we have in the field of epidemiology is to use data to try to get an unbiased and precise estimate of a true effect of an exposure or an intervention on outcome in a population at risk. So, for example, when I talk about population at risk, for example, it might not make sense, to try to understand what are the risk factors for prostate cancer in females, because they do not have a prostate. So we really are interested in trying to understand which population is at risk and how can we understand what causes disease in those populations. And of course, that we are just not interested in sort of theoretical value, understanding risk factors. We also want to use these to prevent and treat disease, or somehow, perhaps in situations when there are screening tools for a particular disease, to try to make diagnosis as early as possible. So that's really sort of why we are here, and we will sort of apply this to genetic epi as this course move along. For those of you who have taken epi courses, you probably spend a lot of time talking about cohort versus case control studies. So cohort studies is sort of the golden standard when you do an epi study. So basically what you're doing in a cohort study is you want to have a population to study your risk factors, right? So you go out in the population, you recruit individuals. For example, I spent many years working in the nurses' health study, which is a large cohort of female nurses that we recruited back in the 70s. So reached out to nurses, sort of thinking that, that would be a good group to assess both disease and risk factors. So you identify your population, you measure all kind of different exposures of these individuals, and then you follow them for time to identify new cases. And so once you have identified who is a case or who is a control, who has a specific trait or not, you can actually go back and look at sort of what were their exposures, 5-10 years earlier. We can try to understand these sort of long-term risk factors. So this is really sort of the golden standard how we do epi studies. And we're going to talk in a minute about what are some of the drawbacks are. But, the obvious one is, of course, that, as a PhD student or wherever you might be, or junior faculty, you might not be interested in starting a course study today and then sit and wait for 30 years to get outcomes, because, we want to publish our papers now, right? So an alternative to cohort studies that's become particularly popular in genetic epidemiology, and I'm going to talk about why, is case control studies. So here you sort of flip it, but you've identified the disease of interest that you want to study, may be type 2 diabetes. You identify type 2 diabetes cases and you recruit them into your study. And then you identify controls that are appropriate for that group. And so when I say, appropriate controls, for example, if you are recruiting diabetes cases that are in their 70s, it might not make sense for you to go and recruit controls that are in their 20s, because those are two very different populations. So as I said, one of the sort of drawbacks with course, even though that is sort of the golden standard in epi and how we sort of been doing things, it really sort of is a very big undertaking, and it's a very long-term and costly approach. So for example, here I'm showing you a couple of examples on how many people you would need to recruit into your cohort if you want to wait about 5 years to have a specific number of cases. So as I said earlier, I'm really interested in breast cancer. So, if I want to have 4,500 cases after 5 years, I would have to go and recruit 1 million individuals from the get-go. And of course, that is a very big undertaking in order to get your 4,500 cases. And as we're going to talk about in genetic epi, particularly 4,500 cases are often not enough to find these sort of low penetrant genetic variants we are often looking for. Of course, one really important pro or advantage with cohort studies is that you don't just can look at breast cancer, right? You can look at a bunch of different diseases and traits that you're interested in, and sort of you're not married to one particular disease type, which you would be in a case control study, because then you actually initially go in and you recruit cases of that particular disease. So case control studies are really, they are cheap, they're fast. And, you know, if you want to have 10,000 cases or something in a case control study, you can actually go out and only, you know, recruit those 10,000 cases and then, you know, 10,000 matching controls, whatever it might be. So, you know, instead of 1 million individuals, you know, you can only go for 20,000 and you still would have twice as many cases than for a cohort. So, so it's really sort of become in genetic epi, case control studies have really become sort of the standard way often how we do our analysis. And we're going to come back and talk a little bit about sort of cohorts and the role they're playing in genetic epi as well in a little bit later. So two very different important concepts in epidemiology and in genetic epidemiology, but I think in particular for observational epidemiology, is association and causality, and sort of the importance of separating these two concepts. So when we talk about association, we say that an exposure and an outcome are associated if there's a differential distribution. So for example, if a prevalence of an exposure differs between cases and controls. Now, just because the prevalence of the exposure differs between cases and controls does not necessarily mean that that particular exposure is the cause of that disease. So this is one of my favorite examples of sort of where you can see an association, but there's not true causal effect. So, you know, you're sitting in an airplane and you get annoyed because they turn on the seatbelt sign so often, because whenever they do that, that will cause it to get bumpy. But of course, you know, that doesn't mean that, you know, putting on a seatbelt actually means that you're going to hit turbulence. So we see association a lot in epidemiology, and sort of one of our toughest tasks and one of the things we need to spend time thinking about, and this sort of goes way beyond, you know, collecting your data and analyzing your data, is try to understand why do we see this association, and do we think that this is actually telling us, give us an accurate estimate of a causal effect. So when I talk about causal effects, what we mean then is that an exposure is causal if the presence or absence of that particular exposure will influence whether the outcome occurs, whether it be directly or indirectly. So so of course, you know, even if you would not put on the seatbelt sign, you will still hit turbulence, right? So they don't. So it's not a causal factor. And again, sort of trying to separate these two are one of the main important things that we need to think about in epi. And one of the things we're going to talk about, which I'm sure if you have been doing any genome-wide association studies, is, you know, one thing we spend a lot of time thinking about is we might identify a lot of genetic loci associated with disease, but what is really the causal variance? Since we have a lot of correlation in the genome, it's a very hard process to go from just an association to identify which of the genetic variants in that particular region is actually causal for the disease. And we're going to come back to that as well. So there's a lot of different reasons why things can go wrong in epidemiology. In general, in genetic epi, and I think this is why I really like genetic epi, we're not struggling with bias as much as we are doing in observational epi. And we're going to come back to why that is. And also we're going to talk a lot about the potential sources there are in genetic epidemiology for bias. But here's just showing you a couple of different reasons why we can get biased associations in our studies. These are by no means exclusive. There are multiple other sources of bias as well. But things like selection bias that I already mentioned. Survival bias is definitely something that we are also struggling with in genetic epidemiology. this happens when, you know, you are recruiting cases after they were diagnosed. So this would be a, you know, case control study. And it might be that if you do that, you might not have been able to recruit those folks that had a more aggressive or fatal disease, for example, heart attack. So actually the cases you're recruiting are a milder case than you would see in the general population. And we have diagnostic bias, which basically mean that whoever is, if you actually know whether or not the person was exposed, can affect how you would assess their outcome. And also a very common sort of issue that can come up in particular in different studies that uses questionnaires or self-reported data is recall bias. It might be that, you know, if you have a family member, specific disease, or if you have that disease yourself, you might spend more time or sort of try to accurately sort of address these questions, and you might remember things a little bit better as well. So recall bias is something, a differential error we can see between cases and controls in our studies. Of course, you know, those things will not be as much issue if you're doing a core study, because then you will collect some of these things before that particular person becomes a case. So sort of the one of the most problems that we see is sort of, which sort of really can, you know, disrupt this this pathway from association to causality is confounding. And so a confounder is by definition a factor that is a risk factor for disease. It's also associated with exposure, but it's not a direct result of the exposure. So it will sort of work on the outcome independent. So I have an example here when you have this three triangle, when you've noticed that there's an association with having yellow fingertips and lung cancer. Now, of course, you know, having yellow fingertips doesn't cause lung cancer per se, but there is a common risk factor for both having yellow fingertips and lung cancer, and that is smoking exposure. So in this particular case, smoking is a confounder for the association we see with yellow fingertips and lung cancer. And of course, you know, confounding can lead to these sort of false positive findings when it will induce an association between yellow fingertips and lung cancer, whatever you might have. And we're going to come back and talk about confounding in genetic epidemiology studies. I think one of the probably the most well-known potential confounding factor is population stratification, and we're going to talk about that tomorrow. So we're going to have our first breakout activity. We are going to talk about confounder. And so this is an example when we talk about birth order and Down syndrome. So there's been an association that's been pretty clear between birth order and the risk of getting Down syndrome. So, you know, if you are the fifth sibling to be born, you have a higher risk of having Down syndrome. So, so we are asking you 3 questions. The one is, can you think about something that would confound this observed association? And once you have figured that out, can you use data? How can you use the data on that proposed confounding factor to reassess this association? And then once you all have thought about those things, I just want to hear a little bit more of your thoughts on what are potential confounders in genetic epidemiology do you think we need to assess? Okay, folks, so welcome back. And so I'm curious to hear your thoughts. So what I am going to ask you all to do is that I'm going to ask you to put in your suggested answer for the first question or a particular confounder in the chat, but I don't want you to push enter until I say go, so we can get all the answers at the same time. So I'm going to give you 10 seconds to put in any favorite confounder you have for this particular association. Okay, go, folks. Age. This is brilliant. Yes, you are all on the same page, which makes it for not as fun discussions, but it's great. And that is the obvious confounder here as well. And just to show you, this is what happened to this association when you are to stratify the association between birth order and risk of Down syndrome by maternal age. And you, as you can see, the association goes away completely. So the other thing I want to follow up and also is to please, and you don't have to do it all synchronized this time, but do you have any thoughts on what our confounders could be in genetic epidemiology? And sort of some of the things that you're all going to have to grapple with when you do these studies. So anything you can think of, please put in the chat. Socioeconomic factors. Comorbidities. Population stratification, genetic ancestry, environmental factors, microbiome, epigenetics, back to ancestry, occupation, geographical location. So these are great, and then we're going to talk a lot about these things, and I think, you know, I think the only comments I'm going to say about this is that I think back in the days, sort of we, and now I speak about myself and sort of genetic epidemiologists, we sort of population stratification was really where we spend a lot of our time thinking about, you know, this is all the confounding there is. I think that the more we have been doing these studies, we realize that there are other potential confounders as well. And so, for example, genetic epigenetic association studies, you know, a lot of the things we're doing sort of, you know, are based on the assumption of random mating. And I think that, you know, that's something that we are recognizing more and more, that this not be particularly true. And we're going to talk a little bit about, I'm going to mention it sort of in passing, but a big cohort, a lot of genetic episodes now is happening is UK Biobank. And sort of it's been sort of recognized now that UK Biobank is a very specific population, and there is often, you know, correlation coming together with genetic variants that are outside the genetic variants, so sort of, for example, you know, different social structures and other things. So that even though they might not sort of be the textbook example of confounder, they can definitely induce associations in close cohorts that would not be there otherwise. Okay, so we're going to shift gear and we're going to leave sort of observational epidemiology and classic epi, and we're going to jump into genetic epi, which is the reason why we're all here. So really, genetic epi is really sort of a section of epidemiology, and here we're interested to understand what are the role of our genetic factors in determining health and disease, both in families but also in populations. And naturally, sort of when we think about genetics and our inherited DNA, family studies come to mind. We are not going to cover family studies, family-based studies in this course, just because of lack of time. If you have questions about family-based studies, I am more than happy to talk to you about those offline or during office hours, because those are obviously, you know, in particular historically, a very big part of genetic epi research. And many of the initial findings that came out in genetic epi were based on family-based studies. I think as sort of tools are improving, we have more resources, we've started to sort of go more towards just population-based research and looking at unrelated individuals. But I'm more than happy to talk about family-based designs as well, if you're interested in that. So genetic epi is kind of like everywhere now. And basically, I think in particular, as I said, there are all these new resources coming. UK Biobank is one, Biobank Japan is another one. 23andMe, which you may not have heard of, a direct-to-consumer company where you can sort of get a swab, you can send it in, and they will send you back, all kind of risk profiles. And so, we have been doing a lot of these large-scale genetic association studies, mostly GWAS, and identify all kind of genetic associations out there. So this is just giving you sort of a flavor of what has been out there so far. And I think that, I just want to talk a little bit about the sort of new biobanks that are coming, because they've really been sort of the driver for our genetic findings over the last 5 years or so. And so the UK Biobank is from UK, FinGen is a Finnish initiative, Biobank Japan is a large Japanese biobank, and all of us is, here in the US, with the goal of recruiting 1 million individuals and link their whole genome sequencing data to health records, electronic health records, and also to self-reported data and physical activity and other things. So all of us, they are still in recruitment phase. So one thing with these, that's fantastic with these resources is that they're making their data available to the community. So you can, write a proposal, and you can apply for the data, and you can get it, and you can run your own analysis. So you don't have to go out and, recruit your own cohort of 500,000 people to study breast cancer. You can just sort of write your application in the UK Biobank and leverage that data. So I assume that many of you have either heard about these, or you're probably also doing some analysis on these data as well as part of your own research. So I would be more than happy to discuss those as well. For example, our group, we are doing a lot of work in UK Biobank and have been doing for quite some time now. So these are tremendous resource to the community that we are lucky to leverage, and it's really sort of driven the research forward. It has also perhaps, I think, also opened up opportunities that does not perhaps allow us to reflect as much about what is the research we're doing, but that's something that we can talk about throughout this course as well. Okay, so why do we want to study how our genome is involved in disease? Do you all have sort of a favorite reasons? I know we talked a little bit about this, about the breakout activity earlier today. If we take 30 seconds or so, if you want to put something in the chat, if you have any thoughts on why do we care? Why are you here? Could lead to better outcomes for patients because it's interesting. Yes, I agree with all this. Understand disease biology, patient-specific, personalized medicine, treat and prevent human disease, and determine causes. So these are all very, very good things, and this is definitely these are definitely like some of the reasons why we are really studying these things. And so the way I think about this is sort of 3 main reasons, and I'm going to give you an example of each of them. So the way I see it is that, doing these studies can be relevant for public health, understanding the biology and the etiology behind disease, and also some obvious clinical applications. So I'm going to go through these one by one and just give you sort of an example. And I'm going to start with sort of public health application. And so this is really coming and sort of trying to identify groups in the population that have a high risk of developing disease. So if I identify those individuals, we can sort of intervene with either specific intervention, but also if there are available screening tools, we can utilize those. So the example I want to give here is the BRCA1 gene. So So back in 2013, and I realized this, that, this might tell you all how old I really am. But there was a quote that came out in New York Times from Angelina Jolie when she talked about, her mom passing away from ovarian cancer in her 50s, and she was carrying the BRCA1, a mutation in BRCA1, and so did Angelina Jolie as well. So she ended up having a double mastectomy and oophorectomy when she removed her both breasts and her ovaries in order to prevent disease. So once sort of this came out, I think it sort of re-established. It was very interesting to see sort of how this sort of became common in the public and thinking about these things. So BRCA1 was detected back in the early 90s through a family-based linkage analysis. And we now have a couple of different genes that we know predisposes you to a very high risk of developing cancer. So BRCA1 and BRCA2 are two of them. Particularly BRCA2 has been associated with multiple cancers beyond just breast cancer. So this is just showing you some of the breast cancer risk associated with carrying one of these mutations. And just to sort of bring back, come back to the fact that there's sort of what we call the Angelina Jolie effect. So she came out with her op-ed in mid-2013, and you can see sort of there was this sharp increase in BRCA tests out in the population, sort of, and the folks that sort of were interested in learning more about their family history and their genetic risk just after, she came out and made her statement and talked about this publicly. So as of today, there is no sort of general testing of BRCA1 and BRCA2 in the population as part of our healthcare. 23andMe, which is this direct-to-consumer company I talked about earlier, they do offer testing for BRCA1 and BRCA2. But one thing that is very important to know, and I sort of highlighted here in this purple box here to your right, is that when 23andMe say that they are testing for BRCA1, BRCA2, they're actually only testing for 3 different mutations. And we know that there are more than 1,000 mutations in these 2 genes that will bring you a high risk for breast cancer. So by no means, getting a negative BRCA1, BRCA2 result from 23andMe does not necessarily mean that you don't have a high risk mutation. And I think that is sort of has been one of the dangers with this type of DTC testing. And there was actually a study came out when they showed that, there was a lot of false negatives in these populations. And sort of this is what you can find now in 23andMe's website. So there's a question about, are those 3 more common than other variants? So these 3 particular variants are common in Ashkenazi Jews folks, so folks of Ashkenazi Jews ethnicity. So it turned out that they showed much more, and you can see they actually say that here too. These are some of the most common in people of Ashkenazi Jewish descent. Whereas, among sort of individuals that are not of Ashkenazi Jewish descent, they see this between 0 to 0.1% of the individuals across other ethnicities. So they're very rare in other populations. So we can definitely use, sort of, screening for these high risk genetic mutations. But as Deanne is going to talk about on Friday, we can also sort of incorporate more common genetic variants with low risk penetrance, sort of, the folks we identify in GWAS now, as in our polygenic risk scores. And sort of we've come to a point in polygenic risk scores, we're not just using, SNPs that we've been identifying in our GWASs, we can actually rely on sort of the whole genomes now, and just, plug in your whole genome using, methods like LDPRED and others to build polygenic risk scores to predict disease. And we will talk more about that on Friday. So another reason why we like to study our genome is to understand biology. And I think this is really where I come from. This is sort of my favorite niche of genetic epidemiology. I really want to learn more about the biology and the etiology. So I want to come back to the example I talked about on causation earlier. And sort of two of the main things that we can partly overcome in genetic epi-studies compared to observed epidemiology is confounding. We can sort of bypass a lot of confounding because, you know, you're sort of born with your genetics, and also reverse causation, which means that, you know, your whatever disease you're having, that's not going to affect your genotype because your genotype will come before your disease. So the example I want to give here is at HDL, which is the good type of cholesterol, and myocardial infarction. So it's been an inverse association between HDL cholesterol and MI for quite some time. So, and this sort of triggered people and be interested in, so can we sort of, you know, if you eat a lot of HDL supplements, will that help you to decrease your cardiovascular disease risk? So that didn't really turn out the way we wanted. And one reason why that was, and how we can study why this is actually happening, is that we can look at individuals' sort of genetically cholesterol levels. So sort of your natural cholesterol levels due to genetics. And we can ask the question, are those associated with your cardiovascular disease or not? So this is a study that came out now 10 years ago, when they looked at genetic variants associated with LDL, which is the bad cholesterol, and HDL. And they saw if you genetic variants associated with high LDL increases the risk for MI. But genetic variants that were associated with high HDL did not increase your risk for cardiovascular disease. And sort of arguing the fact that, you know, we think that LDL is causal for this disease, for cardiovascular disease, but HDL is probably just a proxy for something else. And indeed, HDL is correlated with a lot of things that we know sort of is relevant for cardiovascular disease. Oh, sorry. So the last example I want to give is clinical applications and using sort of GWAS, genetic association studies, to develop new treatments. So rheumatoid arthritis is an inflammatory quitting incurable disease. It's a chronic disease. About 1.5 million individuals here in the US has it. And since this is an incurable disease, people with RA, they sort of rely on having you know, treatments for the rest of their life to sort of handle their symptoms. And the problem is that, you know, after a while, many of these sort of treatments will stop working. You have to sort of switch to the next version out there. At some point, you might actually run out of, you know, potential treatments. So this there was a GWAS performed on RA back in 2014. And what you see here is a result, typical results from a GWAS. So this is called Manhattan plot. So each dot here represents a SNP, a genetic variant. Ali is going to talk more about SNPs in just 20 minutes. And they're color-coded by chromosome. And on the y-axis, you have the minus log p-value for their association with RA. So the higher up you are in this plot, the stronger association we see with this genetic variant and RA. And so as you can see, they found a bunch of things. So the horizontal line here is 510 to minus 8 p-value threshold, which is often what we use to call genome-wide significance. So they found a bunch of like hundreds of genetic variants, excuse me, associated with RA in this study. And when they went back and looked at sort of the genes that were targeted of those genetic variants, they saw that a lot of them were actually already RA treatments, but other of them were actually associated with treatments out there that are for cancer and other things that have not just been used for RA. So in this particular case, there might be potential effort to actually like repurpose those treatments to RA, which would, you know, save you like 10 years or so of drug development. So really, genomics can really help us find these shortcuts and identify new drug targets that we can use to develop new treatments. So this is going to be my last slide. And so I just want to introduce you to pedigrees. So this is a situation where you have a patient, is a 43-year-old female coming in, you're their doctor. They show you their family pedigree. So if you have not seen this before, this is a 3-generation pedigree. So these are her parents, these are her grandparents. She has a significant history of colorectal cancer, both her parents, one of her grandfathers, and also she has breast cancer both with her mom and her grandma. And so she's worried about her cancer risk and wants to know if there's something she should do. So what if you were her doctor, what would you tell this woman? And please put it in your chat and thoughts you have. Genetic testing is one example. Screening. Any other thoughts? So in the US today, the recommendation this person should give was actually not to be genetic testing, because it's not sort of really clear that, that is, what's going on here. Also, she has it both on her paternal and the maternal side. What she should do, though, is that she should start the colorectal cancer screening for family history in US, if you have first-degree individuals, is either 10 years before the youngest diagnosis or at the age of 40. So whichever one of those came first. So this woman should really have had her first coloscopy 3 years ago. And she should also, of course, have regular mammograms. Any questions or thoughts on that? Okay, cool. So we are going to take our second break. We're going to come back again at, let's see, 1:45 Pacific time. So in 12 minutes.
Okay, hi everyone. Welcome back for our last session of the day, where we will talk about types of human genetic variation. So the goals for this session are to describe differences in the types of genetic variation and how they affect phenotypes. And we'll talk a little bit more about the definitions of some of these terminology words. And then to help identify inheritance patterns of genotype-phenotype relationships. So this is really the nuts and bolts of what you need to know about genetic variation and how we can measure actually the genetic data before we want to use that data in our analyses. So this is what we used to have to work with as geneticists and genetic epidemiologists. This is a karyotype. It's the 46 chromosomes that we each have paired up into their pairs of 23. So we get a set of 23 chromosomes from our mother and a set of 23 chromosomes from our paternal side. And they should line up. And it used to be that when we were looking for genetic variation that was affecting phenotypes, this is the level of granularity that we could get. So we would stain the karyotypes and we would look for big missing sections or duplications. And that was the scale of data that we could analyze. But now, of course, we've gotten a lot better with our technology, and we can now look at single sites on the genome to measure. So let's talk a little bit about our genome. So we have 23 pairs of chromosomes. We have about 3.2 billion base pairs in those chromosomes. So that's a lot of information that we could potentially analyze. These 3.2 billion base pairs are organized into about 20,000 genes. And only about 1.5% of all of those 3.2 billion base pairs actually fall within these coding regions. So when we talk about coding DNA, we're talking about the sections of DNA that actually become the proteins that we see. The rest of the information is a little more obscure. And at that point, and the field at this point is trying to figure out exactly what that part of DNA does. But when we think about these coding regions, we'll go back to the central dogma of genetics here, where we're talking about how does DNA become the proteins that we see. And we start with our DNA, and our DNA is translated into mRNA, where each of these sites of the genetic code is basically has a mirror copy in terms of the RNA. And then each set of 3 base pairs is then turned into amino acids that are linked together to form proteins. And these sections of 3 amino acids are what we call codons, these triplets of letters that then link to a specific amino acid in a code book form that can be used to determine what amino acid would likely come next. So the changes in DNA, and this is the DNA sequence itself, is what drives the phenotypic variation. And the phenotype is what we actually see and what we can measure. And this is a trait, a disease, something that we can observe of how that DNA is manifest in terms of a health outcome or an outcome that we care about. So mutations happen all the time. So every time a cell replicates, the genome is also copied. And the human genome error rate in the copying, so this mutation rate, is about 1.1 times 10 to the negative 8 per site per generation. So this is not very much, right? We have a very, very efficient and effective quality control mechanism in terms of our DNA copying. But the human genome is over 300 billion base pairs. So the first activity for this session is to calculate how many mutations do we expect to see every time a cell is copied. If we have a mutation rate that is 0.00000001, but we have 3 billion mutations. So I want you to calculate that quickly and answer in this Zoom poll. All right, we'll do 10 more seconds. All right. So about half of you got the correct answer, which is that we expect 33 new mutations to show up every single time a cell replicates. So that's a lot. So even though we have a very effective replication mechanism, because we have so many sites to copy, there's a lot of differences in the DNA when we're comparing one cell to the next. And when we think about an organism level, we're actually expecting to see about 64 new widespread mutations per generation. So these are variants that we would see throughout most of the cells in the body of an organism compared to its parents. And that's a lot of sites that could potentially be really interesting for outcomes that we care about. So if we're thinking about these coding regions, these sections of DNA that actually become protein, these are easier to think about because these are variants that actually affect the amino acids that we measure in proteins and potentially how a protein works in the body. So we have synonymous mutations, and those are changes in a single site at the DNA, a single nucleotide polymorphism or a single nucleotide variant. You'll see those defined up at the top, a SNP or a SNV. That doesn't actually change what amino acid is chosen. So we often think of these as not being very impactful in terms of phenotypes, because the amino acid that we see in a protein is the same. We also can see a change at the DNA level that does change the amino acid. So you can see here that what we have is a non-synonymous variant, where a change at one site of the DNA changes what amino acid is put in that protein. And this can have a big or a small effect depending on where that amino acid is in the protein itself, whether it's in an active site of where the protein actually does its magic, whether it's at the end or whether it affects the folding of the protein. And then we can have something called a nonsense mutation, which truncates a protein. So if we have a change at one single DNA position, we could lose an entire protein. And again, this could matter where this change happens. It might be at the end of the protein coding region, in which case maybe it doesn't have a big effect at all. Some of these mutations can be more or less bad depending on the type of amino acid that's swapped. So what I've pointed out here is that changing from a leucine to isoleucine in terms of what amino acid is put into a protein sequence maintains the polarity of the amino acid. And so the effect of swapping out that amino acid is less severe than what we might expect for swapping a polar and nonpolar amino acid, for example. So a lot of this really detailed genetic information almost gets into chemistry a little bit of thinking about how do these amino acids interact with each other. And there are prediction tools that will estimate based on where the DNA change happens, so where the variant is, and what type of amino acid effect it has, how severe of an impact they expect to have on that amino acid function. And those have varying levels of accuracy. It's actually really hard to predict what these changes are going to do on a protein level. So these SNPs, these changes at one single base in DNA are what we often measure in genetic epidemiology study. They're the easiest to measure because we can look at one single site of the DNA and compare what base is at a particular location. There's also changes called frame shifts. And this can happen at the level of a single DNA base, where we potentially either lose or insert a single amino or a single DNA base that can shift the entire sequence of these triplet codons. So that you can see here, this figure at the bottom shows that if you lose a single nucleotide, you lose this A, you shift over all sets of those triplets so that each one doesn't quite line up. None of the subsequent ones line up to the same amino acids anymore. An example of this is actually the most common variant that we see for causing cystic fibrosis, and it's the F508 deletion variant. And this is an interesting variant because instead of just losing one nucleotide, it actually loses 3. And so instead of seeing that shift where every single amino acid following this change is different. We just lose an amino acid, and it ends up working out that we end up losing this phenylalanine based on how the codons work. And so this is an example of where we've lost more than one single nucleotide. We've lost 3 here, but you can see that it can have, even losing one amino acid in a protein can have really severe effects on a phenotype that we measure. So the result of losing one amino acid in the CFTR protein leads to a buildup of mucus in the airways that leads to difficulty breathing. So sometimes we don't change the protein itself. And so these coding regions are the easiest to think about because they change the proteins in a way that we can really wrap our head around. But remember, that's only 1.5% of the whole genome. We have the rest of the genome that can also change as well. And these changes can happen anywhere. And if we continue to think about the coding region, so these areas of the genes that are becoming proteins, because that's really what biology is about, our genes end up looking something like this, where we have exons, and these are the portions of DNA that actually become those amino acids. And those sections are divided up by introns, and these are regions of DNA that don't relate to amino acids. They're not going to become the protein in the end, but they sort of divide these sections of exons, these pieces of DNA that are going to be translated directly into protein. And you can see this mRNA here. This is what is going to be used for linking those amino acids together. So the changes in these non-coding regions can also have big effects on that protein formation in ways that don't actually change the amino acid sequence. So we can see here, if we change a variant upstream of these exons, we can change how much of the protein is produced. So we can either have more expression or less expression. And that expression term can relate both to these RNAs and mRNAs that are produced, as well as to the ultimate protein that we can measure. We also have changes that can change the splice site. So remember, as we drop down to these mRNA levels, we have to be able to cut out the pieces of DNA that we don't actually want to turn into protein. And those are the splice sites that we're talking about. So you can see if we change something that tells the translational mechanisms how to link these exons together, we could potentially skip entire regions of the amino acid that we want. So what we're going to do here is do a 5-minute breakout session where I want you to discuss this concept. So there's a recent study that sequenced the genome, and sequencing is where they look at every single position in those 3 billion base pairs. So they sequenced the genome of about 2,500 individuals, and they identified 85 million SNPs. So these are the single nucleotide polymorphisms, changes at one single nucleotide in the genome. They identified 85 million sites where the sequence was different between participants. On average, every single person in this study had 3 to 4 million sites that differed from others. About 0.5% of those SNPs were encoding regions. So these are the sections that actually become protein. And that corresponds to 1.5% of the genome that is in that region. So I want you guys to take a few minutes and talk about why might we only see 0.5% of the variants that are measurable in our population occurring in those coding regions, compared to what we would expect if these variants happened randomly across the population or across the genome, in which case we would see 1.5% of those variants. So we'll spend 5 minutes talking about this question, and then come back and talk about what you guys think. Okay, thanks for coming back, everybody. Let's see, do we have everyone? I think we do. Let's see, so will some willing participants put in the chat what you guys were thinking in your group? related to this question. So why might we only see, why do we see a lower proportion of variance in coding regions than what we would expect by chance? So variance being incompatible with life. Yeah, so that's a really, that's and preventing SNPs from propagating. So we can see that some of these variants that are either incompatible with life or incompatible with reproduction are not going to propagate through generations the same way that other variants might. And so if we think about the severity, so where do we expect the variants to be that would have really severe effects like that is probably in a coding region, because we would see effects on proteins that would make those proteins not work anymore, for example, or do something that would be really deleterious for being able to either survive to birth. So if you think about how many sort of spontaneous miscarriages there are, and how many of those may be caused by these genetic variants that mean that the fetus just can't develop anymore. Yeah, and a term for that would be conserved. So the coding region is more conserved, and that means that what we see between generations is that the coding region is more similar than what we would expect by chance. And this is a survival bias at the very earliest level. So those are all really good thoughts and really nice linking across concepts here. So these single base changes are what we often think about in genetic epidemiology studies. So if we're thinking about these massive platform comparisons like genome-wide association studies, the SNPs are the main unit of comparison there. But we can also have other types of changes here, and these are those structural variations. And if we think back to our first slide of the karyotype, which is where we were looking through a microscope at these macro level structures of DNA, this is what we were really looking at. So structural variation, where we have on the top here, just for reference, is our reference sequence. So the comparison strand of DNA, and we have it divided into blocks just for being able to illustrate the points here. And we can see this, the second row, we have a deletion where that B section is missing. And depending on what is in that B section, we could have some really interesting phenotypes to observe. We can have duplications, which we see on the third line. We can have inversions. So instead of running an alphabet A, B, C, D, E, F, G, it might run the opposite way, which if we're thinking about how whatever coding regions are in that section, and we flip the order, so we, instead of running left to right, we're running right to left in terms of what we would expect that sequence of codons to be, we're going to get a completely opposite amino acid order, or I mean, it's going to be completely mixed up amino acid order if it even turns into a protein at all. And then we can have something that's I think really interesting is reciprocal translation. So when the chromosomes line up, they swap pieces of DNA. And in reciprocal translocation, the pieces that they swap are perfectly lined up with each other. So you can see that there are actually coding regions where you have, the front half from the maternal side and the second half from the paternal side, but they line up so perfectly that it still reads through perfectly. Or you can, and those I think are really interesting. But then you can also have sections that combine with deletions and duplications and inversions, and this can get quite complex quickly for how you measure. So structural variation is a lot more complex to measure, and so we often don't see as many sort of high throughput and big data sets using this type of structure, because it requires things like PCR to measure effectively. But these types of structural variations are still really important in outcomes that we care about. And an example of this are tandem repeats in Huntington's disease, where we see that the more copies of this CAG repeat you have, the more likely you are to be affected by Huntington's disease. And this is a degenerative condition that hits very young. And so you can see if you have fewer than 28 CAG repeats, you're not going to develop Huntington's disease. If you have over 40 repeats, you will develop Huntington's disease. And you can see there's a gray area in the middle where you may or may not be affected. And that's a really interesting position to be in terms of thinking about screening, genetic testing, and implementation strategies that we'll talk about later. So in terms of terminology for how to think about reading and writing in genetics, you often see that we have italics for the gene name. So HTT is the gene, and the protein is written in normal font. So if you're reading a paper and you see italics and you see the same letters in italics or non-italics, one often refers to the gene and the other to the protein. So one quick note I want to make is the concept of germline versus somatic mutations. And often we think about the DNA being the same in every cell in the entire body. And so we often assess our genetic data from tissues that are really easy to measure. So we think about things like blood draws, cheek swabs. We're getting the DNA from those locations and assuming it's the same throughout the entire body. That is mostly true. But if we think about cancer in particular, the DNA in a tumor is going to be different than the DNA in another part of the body that's not affected. And those we call somatic mutations. So those are changes that happened post-gamete stage that are not, so the variants that we detect in those tissues are not going to be widespread throughout the whole body. And I think there's a growing awareness that this is something we need to consider in terms of assessing genetics. So let's talk about how we think about the DNA changes in the context of having two chromosomes that are paired that often have the same types of they have matched genetic information, and then the phenotypes that we actually measure. So because we inherit two copies of each chromosome, we have to think about genetics in pairs, where we have two strands of DNA, and we're considering the genotype on both of those chromosomes. So we have an allele, which is the version of DNA that's at a particular location on one chromosome. So we can have an allele being either the C or the T at that single nucleotide polymorphism. The genotype is the combination of both of those alleles. So the alleles are C or T if we're looking at this position. The genotype is the AC. And because there are two options, because there are two nucleotides at each location, we have three options for genotypes. We can have two versions of homozygous, and that's where the nucleotides are the same on both chromosomes. So we either have two A's or two C's. And then we have heterozygous, and that's where the nucleotides are different on one chromosome versus the other. So for example, AC. Inheritance patterns are important to think about, and this is how the genotype becomes a phenotype. So because we have two copies of every gene or every chromosome, usually, if we think about the XY chromosome, it gets a little more confusing. We have a dominant inheritance pattern, and that's where you only need one copy of a particular nucleotide to see an effect. We have recessive, and that's where you need two copies of that variant to see an effect. And then we have additive, and that's that the effect of one variant is half that of two variants. And the default in many genetic studies is using this additive model. So this is an example of how you might measure the phenotype. So if you have phenotype on this y-axis, and this is, let's say this here is a quantitative trait. And then you have these two options. So you have heterozygous, and this, we often use A and B to fill in for sort of generic nucleotides. You have homozygous A and heterozygote, both have the same level of an outcome compared to the homozygous of the variant. Additive, you can see we have this linear effect. And then dominant, you only need one of these B variants to see an effect. So when we think about how we want to measure and analyze phenotypes, we have two options. One is a categorical outcome, and the easiest here is a binary outcome. Yes or no. Do you have a particular outcome or do you not? Do you have a disease or do you not? The other is a quantitative outcome. So that would be something that is continuous, like height or protein level, speed, for example, or cholesterol level. And the image here shows people lined up by height and wearing a shirt based on gender. And you can see here we have both binary trait and continuous traits. And what I'd like to see in the chat here, if anyone has any idea of how you could turn something like a quantitative trait, so for example, height, into a binary trait, because there are different analytical approaches and different pros and cons that we might want for using binary or quantitative. What might be approaches for how you would turn a quantitative trait into a binary trait? And how would you approach that? So we have this idea of splitting into groups, having a cut point for tall versus short, picking a threshold of some kind. And that's exactly right. And what is important to think about is, what is that threshold going to be, and is it meaningful in some way? And so if we think about the translational side of genetic epidemiology, let's say we have a quantitative trait that's let's say a cholesterol level. At some point, you have to decide who you're going to categorize as having high cholesterol or not. And those are the people you're going to run your intervention on. That's where we have to turn this continuous outcome into something that's actionable. And that's where we would want to turn it into a binary. And often that's done with some sort of calculation of risk level and frequency of the health outcomes that we care about. But it's useful to think about those approaches and when we think about continuous outcomes. So in terms of phenotypes, and these are what we actually measure, so the health status or the level of something, we have something called a Mendelian phenotype, which is driven by variation at a single genetic locus. So these historically were the easiest to study because we only need to find the genetic variation at one specific gene or one specific location in the genome. And we can know, we can make the link pretty directly with the outcome that we're measuring. Where we're getting into more interesting work now is these complex phenotypes. And this is a little more confusing, where we don't have these sort of classic inheritance principles of seeing traits propagate through generations, where we can have oligogenic, which is a few genetic loci affect a phenotype, or polygenic, which is where we're combining information across many sites in the genome to that all together in concert, or a few of them together, and different combinations will affect the phenotype that we measure. So the other piece that makes this all even more complicated is that we have variable penetrance. So we can have the exact same genetic pattern between individuals, but see and measure different phenotypes. And this is a penetrance issue where we have some people who don't don't exhibit the traits that we might expect based on their genetics. One more piece of terminology is this idea of haplotypes. And this will become really important later in the session when we talk about linkage disequilibrium. And this has to do with the idea that because DNA is inherited in strips, so in these chromosomes, they're linked together and each site of DNA is not inherited independently. So what you have for a nucleotide at one position influences the probability of what nucleotide you'll have at another probability. And so sometimes we think of these haplotypes, and those are sets of nucleotides across regions of DNA that are inherited together, and that we see show up together with more frequency than what we might expect if each was being inherited individually. So what we can see in this picture here, we have sequences of DNA. There are only 3 sites that really vary between individuals. And if we're building a haplotype, we'll take the information at each of those sites that actually differs between people, cut out all these regions that are the same across everyone, and talk about the haplotypes, which are the combination of the sites that are actually different between people. So one resource that's really useful to know about as a genetic epidemiologist is dbSNP. And that gives us information about every variant that has been described and named. And if you search dbSNP, it should come up. And I have links to it in the breakout activities. But every single nucleotide polymorphism that has been described has a unique identification number. And that's what we see here. This RS776746 corresponds to one particular nucleotide in the genome that has been shown to have some variability. And this type of reference number is really important because we have, depending on what genome you've sequenced, what the references are, the positions and the chromosomes might not line up properly and be easy to compare. And so being able to use these reference numbers make it easy to compare across studies and across different versions of human genome reference sites. So dbSNP is a really nice resource. It includes frequency information. So how common are these different nucleotides across different populations, if it's been linked to any clinically significant outcome, where it is, what gene it's in, what type of variant it might be. Is it non-synonymous in a coding region? Is it intronic? So we don't have a ton of time here, but what I would like you to do is go back into your breakout rooms for 5 minutes and really focus on this dbSNP. So the activity number 3, which is where you'll gain experience navigating dbSNP and figuring out how to find different information within dbSNP. And we'll spend 4 minutes there and then come back and see if people have questions. Okay, welcome back, everyone. So this variant is in the ApoE gene, which is highly implicated in Alzheimer's disease. And it is a missense variant. So it actually changes the protein sequence of the ApoE. You can see that the frequency was about 8%, although this varies based on population and what study was done and with who, which is important to think about as we move forward. Did anyone have anything that they tried to find on dbSNP that they couldn't? So each of these different tabs that you'll see right under that blue header section will take you, I actually can show you what I'm looking at here, will take you to a different piece of information. So you can see information on the variant. And you can see this is why these reference sequence matter, because depending on what sequence reference is used, the position is going to be a little different. These are also really nice resources for understanding what allele is considered the minor allele. So minor allele is what we consider the least common variant. So you can see sometimes that varies depending on what study was done. Sometimes it's the C and sometimes it's the T. Let's see, what else is worth bringing up here? Publications, you can see the type of publications that use this variant in it, which is a nice way to do your literature searches. And then clinical significance, which is also useful for getting background information. So where does this sequence reference come from? So these are standard builds. So for example, the position, the GRCH38P13, those are reference human genome builds that are used across studies to gather information. And what can you read from the variant viewer? So the variant details, oh, so this viewer that shows the map. So this map here will show you, so this green bar here shows you coding regions of DNA. So you can see that because there's a green bar here, you know it's within a coding region of a gene. So if you hover over that green bar, it'll show you it's in the ApoE gene, and it'll give you details of that gene itself. And then each of these little color blocks show you a different SNP. And each one you can see has a different reference ID number. And if you click on it, it'll take you to that page where you can find out more about that specific variant. So we will end there for today. The goals for this session were for you to get an idea of what type of genetic variation can affect the phenotypes that we measure, and to see that these single nucleotide variants are the most commonly used in genetic epidemiology studies, but that larger sections of DNA can be affected as well. So we'll hang out here I'll hang out here for a few more minutes if you have any questions. Otherwise, we will reconvene tomorrow morning for a few more sessions.
Yes, and we will upload the lecture the whole day today, later today, as soon as that's done. And we will also upload answers to all the breakout activities we had today on the website, so you can access those all there. And we will start bright and early, 8:30 a.m. for those, at least for those of you who are in Pacific time. Better for those of you on the East Coast.